Good morning, everyone. This is from Kristu Jayanti College, and today we are going to discuss about enzymology of DNA replication in eukaryotes and prokaryotes. So there are totally 9 types of different DNA polymerase. In that, we're going to talk about 5 types. Those are alpha, beta, gamma, delta, and epsilon. So the first one is DNA polymerase alpha. It is the largest polymerase enzyme. It has 4 subunits in total, and it has both 5' to 3' and 3' to 5' exonuclease activity, and it has polymerization activity. It is mainly involved in priming activity, only found in nucleus and cytoplasm, and it has 4 subunits in total, right? So it's called as a tetramer, and those each subunits are called as pola 1, pola 2, pola 3, and pola 4. So the second one is DNA polymerase beta. It is the smallest DNA polymerase enzyme found in the cell, found only in vertebrates, present only in nucleus. And the third one is DNA polymerase gamma, involved in DNA replication of mitochondria, present in nucleus, and it has total 2 subunits. So because of that, it is known as dimer. 5' to 3' polymerization activity is present, and 3' to 5' exonuclease activity is also present, and it is encoded in nucleus of the cell but not in cytoplasm, and it is only found in nucleus. So the fourth one is DNA polymerase delta. It is the main replicating enzyme present in nucleus. 2 subunits in total. So basically called as dimer. It has 5' to 3' exonuclease activity is absent, involved in synthesis of lagging strand. DNA polymerase epsilon. So this is the last type of DNA polymerase present in nucleus, and it has 4 subunits in total. So basically it's a tetramer. Those subunits are called as a POLE pole 1, 2, 3, and 4. And each of them has a specific activity and play their own specific role. And it is included in synthesis of leading strand. So as we consider DNA replication, there are a series of enzymes that are involved, such as helicase, SSB proteins, topoisomerase, and many more. So we can discuss about them one by one. So the helicase is the first one, which helps in unwinding the double helix. At O-ring, it converts the double-stranded DNA to single-stranded DNA. In eukaryotes, it is known as tag 1 and tag 2. So helicase, it is in prokaryotes. And SSB proteins, SSB stands for single-stranded binding proteins. In eukaryotes, called as replication factor A, RFA. So it basically prevents renaturation of the single-stranded DNA to double-stranded DNA and helps in stabilizing a single-stranded RNA. Next one is topoisomerase. There are 2 types of topoisomerase. Those are topoisomerase 1 and 2. This releases the topoisomeric stress from single-stranded DNA. So next one is RNA priming. It basically helps in synthesis of RNA primer. It works with DNA polymerase alpha. And the next one is DNA ligase, which basically seals the gap and helps in formation of phosphodiester bonds. The next one is flap endonuclease. It involves in removal of RNA primer after the end of the replication. And the next one is replication factor C, which basically acts as clamp loader, and it assists in DNA polymerase delta to form a clamp between DNA polymerase alpha and pCNA. Thank you. Please like, share.